Hello, all. Let's see. I know JP said he was going to be late, and I don't know if Megan was planning on joining us or not. So we'll wait just a second for her. Caitlin, I don't know if you know if Megan was planning on joining us. Don't know. Okay. So I want to respect your guys' time and mine as well, so that we're not all up too late. I figured we could spend an hour. We could talk about stuff. Brenna, you had asked, I think in a message to me, and I figured I'd address it tonight. You'd asked, is there a way to like post a key for the homework? And yes, I will do that. I realized that my key, because I made a key a few years ago, and then I've moved problems around and I've changed the wording of things, and it was actually, I was worried about it would be too confusing. So what I think I'm going to do is I'm going to use, this is actually a student from last year. I'm going to use their homework as a way to kind of walk through. I'm going to kind of show it on screen, and we'll sort of walk through it, and then I will post that one so we can see. And that will wait as I go through it. I'll also make sure that there's not any errors in his homework that I post for you guys, but we'll do that. We'll go over the homework. I'm going to sit there with my, with the exam open, and I'm going to kind of highlight a few things that I want to make sure you guys study. I'll give you sort of an overview of what, like it sounds weird. There's only 5 questions, I think, but the first 2 correspond to 66% of the exam. And they're sort of like how you've seen before. They're like just like a little blank, provide one answer for something like, what is the enzyme that does this chemistry or whatever. So we'll kind of go through that, but why don't I start with, let me pull up this homework file, and then we can just go through the homework. In particular, if there's something where you're like, I might go quickly, and then if there's something you want me to explain a little bit more, but I think sometimes the problems are pretty straightforward.
Can I ask a question to start off real quick?
Yeah. Mm-hmm.
Could you reiterate how pyruvate regulates glycolysis? I know that was on homework and stuff, and we've talked about it quite a bit. And I have like different jumbled notes, but I'm having trouble like piecing it all together.
So I don't know if I've talked about how pyruvate regulates glycolysis.
Not pyruvate, PFK. I'm sorry.
Oh, okay. Sorry. Okay. So PFK is the rate controlling enzyme. Okay. So if we want to think about how it's regulated, and for right now, the only way that we talked about for what you need to know for the exam, and this could be why this is getting confusing, because we sort of like elaborated on it more in our glycogen metabolism chapter, which is not anything you need to know. So what you need to know is, I'm going to open up the homework because it is one of the homework questions that kind of relates to it. So let me open this up here, and I'm going to share my screen. All right, so do you see this homework sheet here? Okay, so this is a student from last year, but I'm going to get to the question that kind of talks about regulating PFK. Well, actually, there's a couple of questions here. So why don't we go through this part of the homework, and even though it's a little out of order, we'll go through these questions. So PFK is regulated by structural allosterism. So explain what this means, okay? So structural allosterism is basically, to define it, it's binding a small molecule that regulates whether you're R state or T state. Okay, so let's see what he says here. This means that PFK is not regulated by phosphorylation, which is true with phosphorylation chemistry. He says covalent bonding. That's called covalent modification. So I'm just kind of highlighting things here that are a little bit not correct with his answers here, and is only influenced by secondary molecules binding or orienting a separate part of the molecule other than the active site. So that's true. So you're going to bind a small molecule that changes whether you're R state or T state. And changing whether you're R state or T state is going to determine whether you can bind substrate, R state, or don't bind substrate, T state. Now, a lot of the other enzymes that we've started learning about are regulated directly by covalent modification, by phosphorylation of a serine or threonine. That is not what happens with PFK. But we started learning about how phosphorylation states. Hi, Charlotte. I'm going to say hi. Charlotte's got her suit on. She's chasing Larry. He's not happy. Larry just wants to kind of like lay down and sleep here. So we started learning in this learning unit that there is a way that phosphorylation layers on, but I'm not even going to like be concerned about muddying the waters here with that stuff because we don't need to know it for this exam. Okay. So we regulate PFK activity by structural allosterism. So let me jump down here to figure 12 here, which sort of describes the structural allosterism. So this figure here, right, shows two X-ray structures of the major allosteric changes that occur in a PFK. So T state is shown in blue, R state is shown in pink. Major changes that are seen are in this T to R transition. So I'm going to zoom in here. So this T to R transition that swaps out a glutamic acid residue for an arginine residue. Okay. And we should be able to just predict, okay, if the substrate that I bind to do chemistry with PFK is F6P. I know there's like so many like letters and numbers and abbreviations with things here, but those words make sense. The substrate that we bind for PFK to do its chemistry is F6P. Thumbs up, everybody good? So F6P, especially in this orientation, has a negatively charged phosphate group that's right there on the 6th position. Okay, so what we have here is we've got one conformation of PFK that puts a positively charged arginine in this position, and then another conformation that puts a negatively charged glutamic acid residue there. Now, I told you what the pink and blue sort of related to, but we should be able to predict here which is R state and which is T state. Okay, hopefully it makes sense to everybody that the R state is going to be when the arginine is here, because that's going to form a favorable electrostatic interaction with the substrate. R state means I'm going to be favorable to binding substrate. T state is going to be when this glutamic acid residue is here, because that's going to be unfavorable for binding my substrate because I've got the same charges there. So it's going to be an electrostatic repulsion. Does that make sense? Okay. So let me zoom out here. And I'm hoping like I'm getting like a spinning wheel. Ooh, that's way too far, way too far. Spinning wheel, it was not happy. All right, let's zoom back in here. All right. So noting the position of the substrate, explain how this affects the enzyme activity. So I'm going to read his stuff here and make sure that what he has is right. So glutamic acid 161 is negatively charged and doesn't orient the negatively charged F6P substrate, then considered in the T state. So that's right. While when ADP orients PFK arginine 162 becomes, I don't know. So I don't know what his grammar is here, but when we've got PFK having arginine 162 there, when we interchange that, we now will favorably attract F6P to the active site and now can do chemistry on it. Okay. And that chemistry that we're going to be doing by PFK, didn't need to say that here, but that chemistry that you're doing is you are phosphorylating it, which that's not important to say here because we don't even like see the rest of the enzyme active site that shows where ATP is bound there. So best, does that help with PFK? So that's how we regulate PFK. Then there was just another chart here that just kind of showed us. I will tell you, there is a question on your exam that is for a fictitious metabolic pathway. Okay, so you're going to need to apply things that you've known, but to a system you've never seen. Okay, it's a fictitious pathway that has like enzymes A, B, and C. And I show you a plot and you have to interpret things. Okay, so we're going to go through a couple of those here, but big picture things. When you see something where a curve shifts to the right, that means you bind your substrate more weakly. Okay, which means you're going to have a lower level of activity. So again, we can draw horizontal and vertical lines here. So let's imagine this vertical line here, this dotted line, we're saying, hey, at this same level of substrate, the green line shows that I've got a lower level of PFK activity because we're in the presence of ATP, which is an inhibitor. Okay, so JP and I were actually talking about this afternoon. So I'm going to kind of revisit that conversation that he and I had to kind of explain it, because he's like, wait a minute. So ATP inhibits glycolysis. And this wasn't his words, but as he and I were talking, this was sort of the summary of what I gave him. All right, ATP, we think of as a fed state signal, right? But ATP inhibits PFK. And if we want PFK to be active, well, I should say if PFK is active, remember we talked about glycolysis being a fed state pathway. So there seemed to be a little bit of a disconnect there. Okay, so I'm going to say that again because it was a subtlety that JP is one of those students who often brings up these little things that it's like, yeah, gosh, when I make an oversimplification of things, that's inconsistent. So let's make sure we kind of highlight this. ATP inhibits PFK. That's a little bit counterintuitive because you're like, wait a minute, glycolysis is a fed state pathway. So shouldn't it be active under these fed state signals? But there's two things that are going on with glycolysis. If we are in a normal sort of fed state scenario, we do want glycolysis to be not ramping up too much because we've got enough ATP. But if we are not only in sort of a fed state situation where we've got plenty of ATP, but we have an excess of glucose, we still have to make sure that glycolysis is active because remember, high levels of glucose are toxic. The only way to sort of get rid of it is to pull it into a cell. And at this point, it's going to be liver cells, pull it into liver cells, take it through glycolysis, end up making it into acetyl-CoA so that we can make it into fat. So what can happen is even in the presence of high ATP levels, and this is not data that's shown anywhere here, but if you have high levels of glucose, we're going to keep glycolysis active because it's the only way that we have to get rid of those high levels of glucose. So does that make sense? And it might be something, I'm going to post the video for this. So even if it's something that you're like, I sort of got what Dr. Parenti was saying when she was talking about the ATP and glucose thing, but you can go back and rewatch it. Okay. So let me just come back to this graph for a second, because I want to make sure you guys understand looking at these graphs, right? If we keep this substrate concentration the same in the presence of an inhibitor like ATP, what we're going to see is a low level of PFK activity. Look at the y-axis, right? That's phosphofructokinase activity. It's a low level of activity. When we have no inhibitors and low ATP concentration, we have a high level of activity. Okay. Now, if we draw a horizontal line and say, you know what, I want to maintain the same level of activity, what we need to do with the green line is have a higher concentration of substrate. It's like, hey, the conditions are telling me with ATP, don't be very active, but I can override that by having a high concentration of F6P. Well, how do you have a high concentration of F6P? You've got glucose being made into glucose 6-phosphate being made into fructose 6-phosphate. So that's sort of how we can override that, ATP level, is if fructose 6-phosphate starts getting really high, look at, we can still get to the same levels of PFK activity. Gosh, that was a lot, but does that make sense? Questions? All right. So let's see, based on what you know from the answer above, explain the green and the blue data. So I think we explained that, but let me see if his answer is right here. The blue line is the activity of PFK without ATP, which suggests it is, nope, he's wrong here. Maybe I'll just post my key. Let me see if I can find it here, because I don't want to post something that's wrong. And it might just be, you'll find the same questions. They're just going to be different numbers and they might be in different order. But I can still, let me see if I can find it here easily, quickly enough. Maybe I have it here. I don't know if I made a PDF copy of it. Okay. Let me see if I have a PDF, and if not, I'll just quick make a PDF. Now I'm just going to do that right now. I'm going to quick make a PDF and I'm going to post it. So anybody have questions real quick as I'm like scanning my PDF here? I can multitask and talk and do this at the same time.
Hey, Dr. Penny, I just had a question.
Yeah.
So are we gonna have something on the test where we have to explain active site or no?
No, I will tell you that you don't, but you do have, there's 2 questions that involve mechanisms. So I want to make sure to talk about that tonight a little bit, make sure that you guys, think about how to think about mechanism. So you do want to make sure that you know your structures of molecules that are in glycolysis. So even if you're not like even if you miss parts on a mechanism for something, if you got the starting and ending structures right, you're going to get, at least, half credit probably for that piece. So, but I just kind of wanted to highlight that, I do want you to know mechanism.
When you say know the structure, do you mean like know the structure for some of the bigger things that we've learned? Or do you mean like the structure of like DHAP or like
Yes.
something kind of
Yep.
like
Yep.
everything?
So
Okay.
every structure of a substance that we talk about in glycolysis, I want you to know the structure of. So 1,3-BPG and 3PG, 2PG. So yeah, I mean, and that is, so again, I'll show you sort of the beginning part of that question in just a second here. So now that I've finished this, hopefully this is gonna look good enough, and then I will post this after we sort of talk about it here. All right, let's open this. So again, we're going to have a number of the same questions here. They're going to be different numbers because I've gotten rid of questions over the years and so forth. So let me open up a file here. All right, so I'm going to start sharing my screen again. All right, can you see this Word file? Okay, this is the exam here. I'm showing you the first question because I want to have you see how this is set up. So the first question actually is worth 28 points. So it's a little more than a quarter of your exam. It's all about glycolysis. Okay. So it says, answer the following questions about glycolysis. Note if I'm asking for a step number. So you'll see that I'm asking for a number, an enzyme name. Okay. So you'll see enzyme in parentheses, or substrate, product, or cofactor names where you'll see SP or CF. Okay, or a chemical structure. So for example, and I'm showing you what the first question is, it says ATP occurs at these steps. And right now, and I'm not going to change it, this will be the first question, but it says enzyme. So tell me the name of 2 enzymes that are responsible for producing ATP. So you might see another question. I'm not going to have the same question about the same thing, but the question could also say ATP production occurs at these steps. Then I'd put a number in, and then you'd say 7 and 10. Okay, where here it's going to be your enzyme names. Okay, does anybody want to take a stab at what are these enzyme names?
I could.
All right, go ahead, JP.
So pyruvate kinase is step 10.
Yep.
And then it's a 1,3-BPG it like removes a phosphate, makes 3PG, and then the isomerization is right after. But can
Well,
somebody else
careful.
help me out?
It's not an isomerization that happens afterwards. It's going to be a mutase enzyme because we move a
Oh,
whole
yeah.
function group. Yeah.
Functional group. That's right.
Yep.
I think it's a kinase. It's a phosphoglycerate kinase or something like that.
Yes.
Is that right?
Yes.
Jeez.
So, all right. So one of the things that I would recommend people do if they're like, oh my gosh, like I do not know stuff to that level. What's
Can I
up?
borrow your charger? Mine's not on.
What charger? My
iPhone.
yeah.
Okay. Because it keeps connecting. Mine keeps connecting and then disconnecting.
Okay.
So you can take it back when you go to bed.
Okay. So here's a study tool, and I know JP says he's been using the glycolysis game stuff, so maybe that's been helping him. But remember when we did, and this is only 16 minutes long, but this is when we sort of walked through glycolysis. So because that one day that we were doing it, it was like we had a Zoom class. And so I was sort of walking you guys through glycolysis with a video here. So it shows the structures, and you need to have it as like a story in your head so that when I'm asking you a specific question about it, you can answer that question. Okay, so that is this first question. So there's actually 25 blanks. Okay, so this happens to be blank 1 and 2. Each blank is worth 1 point. So all kinds of questions, from naming enzyme names that do this, naming substrates, and then there's a 3-point question for drawing the structure of a substrate. Okay, so that is the first question. Then there's another question that's called beyond glycolysis. So after we learned all those details of glycolysis, then we moved on and said, okay, let's talk about some big picture things. And so some of those homework questions are like, what are the possible fates for pyruvate? So I might say, hey, we've learned about, 3 different enzymes, or tell me 3 different enzymes that do something with pyruvate. Tell me the name of the enzyme and tell me the product that they make, right? So those are some of the kinds of things. So let's go through here, and I'm going to have this copy so I can see what question number it is, and then I'll sort of find it on mine. Okay. Any other questions on that before? I'm just going to go through the homework. I think the homework's going to be a good gauge for things that you need to know to answer some of these questions.
Dr. Brennan?
Yeah.
So I was going to ask, the way I learned all of the substrates and coenzymes and everything was very much through the chart on the homework, kind of filling it out sequentially. I was going to ask
that one.
If I white out where it says glucose at the beginning and pyruvate at the end, can I print it out and get there 5 minutes early and fill that out and keep it with me during the test?
So can you have this with you during the test?
Can I bring in a blank one 5 minutes early and fill it out? Because when I think of step 6, I think of step 1, 2, 3, 4, 5, and then I'm okay, 5 produces 6.
Got it. Right.
Just an offer, just a thought.
Yeah, let me think about that, because I need to make sure that that's something that I could offer to everybody. And I don't know. So let me just think on it. I will let you know before the end of the night tonight. But I got to think about that, because I know sometimes that's what students are like. I have it in my brain. I got to get it just down so that I could then do something else. And it might just be if that's what you want to take time doing. Because I just don't know if everybody else could to give some extra time. So I gotta think about it. Because certainly you could start the exam and say, hey, I'm going to take a blank part of the exam and start writing that all down. But
which I might do.
Yeah. So I don't know. I gotta think on it. I'm not going to give an answer right yet.
Okay.
Let's see. So a little bit of math here. I think a lot of people sort of are okay with that math. What is A1C? So A1C is that hemoglobin A protein. Specifically, that number refers to the percent of this hemoglobin that is glycosylated. Chemistry is that you have the N terminus that forms an N-glycosidic bond. So that's what you do with that. So what I'm going to do is I'm just going to read through these answers. And if there's one that somebody wants me to explain a little bit more, we'll go over it. How's that sound? Good. Top down and bottom up approaches for remedying low blood glucose. Top down is where we're going to have glycogen as endogenous starch, or you can have dietary or exogenous starch get broken down, and that's one way to remedy blood glucose. Or the bottom up approach is gluconeogenesis, where we take some non-carbohydrate precursor such as alanine. Remember, alanine is the cognate amino acid that will make pyruvate, right? If you deaminate alanine, it makes pyruvate. Okay. And please, because I can't see you guys too well, just unmute yourself and just like, if you want me to explain a little bit more. I'm not going to go through the fundamental stuff because I think you guys know what you need to know for that. And you took a quiz on it. So I'm going to skip through that. Again, this is a great table, as Brenna was saying, to kind of like know all your stuff. But I got to think about whether or not like having some extra time to sort of write that beforehand makes sense. And that table is also available on Blackboard, like as a standalone sort of answer. So you could sort of make sure that your stuff's right there. Possible fates of pyruvate. Remember, what we do with pyruvate depends on whether we're under anaerobic or aerobic conditions. So the oxidation sort of state of the cell, I shouldn't say oxidation state, the aerobic state of the cell. Where if we're under aerobic conditions, we're going to go through the citric acid cycle and make acetyl-CoA and carbon dioxide. And if we're under anaerobic conditions, then we're going to use homolactic fermentation and lactate dehydrogenase as a way to make lactate. And again, the purpose of all of that is to regenerate NAD+. So that's why this is going to be happening, is it allows us to regenerate NAD+ so we can continue with more glycolysis. So let me make sure that I'm going to try my best to sort of follow along with questions that you guys have. Next question is possible fates for lactate. Sort of what happens with lactate depends on whether we are dealing with lactate in a rapid and local level, where you just reverse lactate dehydrogenase, make it back into pyruvate. And then again, if you're under aerobic conditions, then you're ready to proceed down this pathway. However, if you maybe aren't under aerobic conditions and you need to sort of deal with this metabolic waste, if you will, you can export it into the bloodstream. And that's why athletes will talk about having high circulating blood lactate levels. That's indicative of them being very active under an anaerobic sort of activity. Then it's exported to the liver. This is going to be happening when you have an extended sort of period of time where you're producing lactate. It's handled on a systemic level. So you've got to circulate this out so that it can get to your liver. And then the Cori cycle will remake it into glucose. And then that glucose can get recirculated out into the bloodstream or handled with as appropriate. So that's lactate. Decarboxylation of pyruvate. Okay, we actually looked at this mechanistically. So I would say make sure you know how to do a mechanism for a beta keto acid decarboxylating, as compared to looking at an alpha keto acid and why that doesn't happen. Okay, so when we looked at this, and I don't have a picture for it here, if you have an alpha keto acid and we try to decarboxylate that, remember, anytime we do a decarboxylation chemistry, you're going to be taking a carboxylic acid functional group and trying to fold it into carbon dioxide and then have that break off of your molecule. So I know we have that sort of written in notes. And the problem is if you have an alpha keto acid, you can't actually do that chemistry.
Could we actually review that? I'm a little confused on like
the mechanism.
Well, I don't. Is it? Sorry, I'm closing my garage door. I just got back from work, but I was just wondering if it was in the notes. If it's in the notes, we don't really have to go over it. But
well, it is. But I can find it here, I think. Well,
I
actually,
think that the main mechanisms in Unit 3's notes.
Oh,
well, hang on. Let me look here. Let me look at Unit 2 notes. I'm just going to open these here because I don't know where they are on my computer. My computer is a bit of a disaster right now. Okay. So, come on. Oh, it's just, there we go. All right. So, here we go. So it's page 25 here. So if we look at, here is a beta keto acid decarboxylation. So you see how you have this CO2 group, and I didn't draw out all my lone pairs on this oxygen, but a lone pair of electrons on the oxygen is going to come down and form a pi bond here. Can't have 5 bonds to carbon. So this bond is able to be broken because we can resonance stabilize that negatively charged intermediate by pushing those electrons through to the oxygen. Does everybody see that mechanistically? Okay, so that's how a beta keto acid would be decarboxylated. Now, if we try to do that same kind of chemistry with an alpha keto acid, look what tries to happen. We'll do the same thing. We're going to fold those electrons down to make a pi bond here. We can't have 5 bonds to carbon, but if we try to break these electrons or break this bond, we can't put these electrons on this carbonyl carbon, because not only is that an electrophile, but there's no way of stabilizing that. So that's not going to happen. So does everybody sort of see that situation? JP, does that help answer your question?
yeah. For some reason, my head was going to like alpha and beta glycosidic linkages.
Oh,
So
yeah.
I was.
Nope. You
my
were in
head
a
was completely-
different plane.
Yeah, it didn't look the same in my head.
So that addresses why is the decarboxylation of pyruvate problematic. So how is it remedied? Well, we use vitamin B1, and we didn't go through a lot of the details, but it's what we call an electron sink. You're actually going to have this B vitamin actually serve as a repository for where you can have those electrons kind of get stabilized. And so that's why we call it an electron sink, but we didn't look at anything mechanistically about how that happens. Okay. I think this was a question I took out. You guys don't have this question. So this was the next question here that you guys have, which we went through this, knowing that ATP is a substrate and an inhibitor. And we sort of talked about why it's an inhibitor, but it can be overridden by excess glucose concentrations. Structural allosterism. This is a question you guys don't have anymore. We talked about the blue and green data. This is a question 14 you guys don't have. And we went through this question on the structural piece here for why this is able to affect enzyme activity. Let's see. What's the next question that we have here? We know that FBP, JP, you and I think talked about this question the other day. We know that FBP is converted to F6P. This reaction is catalyzed by the enzyme fructose 1,6-bisphosphatase FBPase, and that has a large negative delta G. Is this consistent or inconsistent with what you'd think about for the delta G for PFK, which is essentially the reversal? So, and again, when JP and I talked about this, it's like, well, wait a minute, that's initially inconsistent. We're going downhill both ways, but that's okay, because what we're doing is we're saying those interconversions are using a different enzyme and a different pathway. So forward direction for glycolysis is PFK, reverse direction for what we now know as gluconeogenesis is FPBase. So we're not inconsistent as long as we choose a different enzyme and pathway, and we ensure that one pathway has the input of energy. And as we know, that input of energy comes in step 3, which is PFK chemistry, which is requires the use of ATP. Okay, so that was that question. And I think I have some questions that you guys don't have here. We don't have that one anymore. And I think the questions that you guys have are much more simplified than what I have in this earlier version of the homework. So yours says stage 1. Let me actually go back to, this one. And we'll just go through the answers here. Oh, wait, let's see. Did I skip something? Nope, that's the wrong homework. This guy. All right, pentose phosphate pathway has 3 stages. So for each of these stages, I want you to be able to think about like what's happening. So stage 1, if the pentose phosphate pathway encompasses the first 3 steps, what name do we use to describe this stage? We said that was the NADPH production phase or stage. What is the main purpose for NADPH synthesis? This molecule can be used to reduce glutathione back to, or glutathione in its oxidized form back to glutathione. Can also act in the synthesis of other molecules, anabolism. What did I have here for mine? I just want to make sure that we're not missing something, because I think I have some of these. See, I used to have a lot more detail and I've taken it out. I know it's probably hard for you guys to believe, like there used to be more details that you had to know. No, I think what he has is good here. That the only other thing that I'd say is it's the purpose for NADPH. We learned it for glutathione regeneration and for lipid sort of synthesis, but it's basically lots of anabolic processes. We're going to be building bonds. Rate controlling enzyme is G6PD. This stage is shown to be irreversible. Why do you think this is? Remember, anytime we talked about removing carbon dioxide, that's going to be a hard step to reverse. Okay, so that's stage 1. Stage 2 is steps 4 through 5. What name do we use to describe this process? Here's the word that I want you to know: restructuring, right? So stage 1 is NADPH production. Stage 2 is restructuring. The end molecules are R5P. This is ribose 5-phosphate and xylulose 5-phosphate. So XU5P. One is used for a very important purpose. So ribose 5-phosphate is used for nucleotide synthesis. Okay, so obviously that's when we're doing a lot of cell division. Stage 3 is step 6 through 8. We call that recycling. Okay, so those are the words that he has more here than you need to know. It's just I want you to know that that stage is recycling. The end molecules are GAP and F6P. What are the fates of these end molecules? They can be sent back into glycolysis to be used as fuel. Absolutely. This stage is shown to be reversible. Why do you think that was important? So that we can siphon off GAP and F6P from a glycolysis and then use this stage to turn them into ribose 5-phosphate if we need nucleotides. So it's the idea that, hey, if I don't need NADPH, I just need some ribose, I can just siphon up from the bottom to do that. So that's the pentose phosphate pathway. Okay. Questions on the pentose phosphate pathway?
Yeah, like which steps go in which stage? So like the first 3 steps are in stage 1.
No, you don't need to, like, if you forget that this is 3 steps and this is 2 and that's 3, that's not a problem. You don't need to know that. But I do want you to know that the purpose of that first stage is NADPH production. Rate determining enzyme, this is the only enzyme we learned a name for in the pentose phosphate pathway, is G6PD. And we learned that name because we understood that it's kind of the most common metabolic sort of disease is a dysfunction in G6PD. Does that help answer your question? So you will not see, the only place that you're going to see like really intense details is glycolysis. Okay, I'm not going to ask you like, what's step number or what's the substrate name or things like that. Like I do want you to know, ribose 5-phosphate. If you don't remember xylulose 5-phosphate, I don't think I have a question on that, but ribose 5-phosphate is one of the main products of the pentose phosphate pathway. And we already need to know GAP and F6P from glycolysis anyway. These are the endpoints of the pentose phosphate pathway if we're recycling everything back. Other questions? I think this was a question that you had, and we talked about this in class a good bit. We studied the molecules, shown it right in detail in previous chapters. We've talked about glutathione a good bit. What purpose have we discussed previously? So the purpose of glutathione is to counteract oxidative species that may harm the body by reducing them. Actually, it should be oxidizing them. I'm going to go back to my key here, just in case he's got, I'll post both of these so you can kind of see both. So I guess I don't even have that many details, but the purpose is glutathione reacts with and just neutralizes oxidative species. So if there's an oxidizing agent, okay, that's something that is going to, I shouldn't say it's an oxidizing agent. It's you know, an oxidizing species that's going to damage RNA, DNA, proteins. You don't want that stuff kind of messing around in your cells. JP had asked me earlier today, he's like, well, what kind of things are those? And I said, well, they could be, you know, oxygen molecules, superoxide radicals, just things that we call reactive oxygen species. All of those things are no good in your body. So glutathione works to say, hey, don't oxidize all of that good stuff in my cell. Oxidize me, make me into this form of glutathione. Now, it would be really wasteful if we used this, if this was a once and done kind of molecule in your body. So what's not specifically shown here in my key, I think Hayden had it over here. Well, actually he doesn't, but the purpose of this is we can take glutathione and make it back into free glutathione in its oxidized form and make it into its reduced form by NADPH. I probably actually have that here. No, where was I going to see that? Here. Sam, you have a question?
Yeah, did I give you my computer or something?
Did you take in your computer?
Yeah, I gave you my computer.
Yeah.
Where is it?
I don't know. I think it was on the counter when I was using it. Remember, I was looking up something on Harbor Creek's website. No, it's on the counter by where Dad normally puts his computer. Yep. Yeah. So here's the nuts and bolts of it. Is that NADPH is responsible for regenerating glutathione in its active form. Okay, so that's kind of what we are doing there. And then we sort of talked about how this interplays with disease. So I want to kind of go through this, make sure everybody sort of understands that there are a few questions that ask you to sort of put pathways together. Okay, so this was one of those pathways that we talked about. And my mistake here, this is just G6PD, not DH, okay? But when somebody is G6PD deficient, that means they're going to have low levels of pentose phosphate pathway activity. If your pentose phosphate pathway activity is low, you're not going to be able to have sufficient glutathione levels. And that's partly because you're not making enough NADPH to regenerate your glutathione, okay? When you have low glutathione levels, you're subject to greater damage by oxidizing species, and that leads to increased red blood cell lysis, okay? And that's by definition what hemolytic anemia is. You've got low iron, low red blood cell levels because those cells are lysing, okay? And I think part of your question is kind of highlighting why do we see this gene, this maybe a malfunctioning gene of G6PD, like very prevalent in malaria regions that also happen to overlap with sickle cell regions is, well, you know what, if cells are more prone to lyse, they're more resistant to malarial infection. So anything that is going to make a red blood cell more resistant to malarial infection is going to be a gene that's kind of selected for from natural selection. So sickle cell anemia, and now we're seeing G6PD deficient individuals actually have a selective advantage in regions where malaria is endemic. So does that make sense to everybody? I'm going to go back because I know we had this on a slide here that kind of showed that same information. So if you want to kind of highlight that that's where this slide was, and then it's also kind of summarized here in this slide. All right, I think this is the last page for your homework. So we'll come back here to my slides and make sure that we talk about a slide like this. So again, you've got a couple of slides here where I want you to know how to read graphs. Okay, not specifically regurgitating this information, but if I gave you a fictitious metabolic pathway that you could understand sort of what's going on here. So I'm going to talk about it in the context of this pathway, but be able to think about what this means. So this molecule 2,3-BPG, where have we seen this guy before? This is responsible for hemoglobin function. So its purpose is to increase oxygen delivery to cells and tissues. How it does that is it favors the T state, right? So it binds to and stabilizes the T state. That effectively increases the P50 of hemoglobin, which means it doesn't hold on to it as tightly. So it's releasing more of it to the tissues. Okay. So how we synthesize 2,3-BPG. So it says this molecule is separately synthesized by siphoning off an intermediate from glycolysis rather than stealing the intermediate from the PGM reaction. So here's figure 5. Let me zoom in here, right? So if we look at what happens with the PGM chemistry, right? That's the chemistry that JP was talking about that interconverts 3-PG and 2-PG, okay? Where we basically are taking 3-PG and throwing another phosphate on. This is a unique kind of role for histidine here. And we get 2,3-BPG as an enzyme intermediate here. Now, we talked about why we don't siphon this off from this point, because that would render this enzyme inactive, right? Because it would not have the required phosphate on that histidine. So even though 2,3-BPG is an intermediate in a glycolysis enzyme that we studied, that's not how we're going to make it. Here's how we make it. We actually are going to siphon 1,3-bisphosphoglycerate off of glycolysis. We're going to use a mutase enzyme to move that phosphate from the 1 position to the 2 position. And that's how we generate 2,3-BPG. Okay. So now that we can see where 2,3-BPG is made, it's made after we've made 1,3-BPG. Okay. But it's before we get to the end of the pathway. Okay. So I'm going to make sure I want to be able to see everybody here. Let's see. All right. Everybody give me a thumbs up if that makes sense to you. Yep. Okay. So now what happens? Let me see. Can I move you guys? No, this way. There we go. I wanted you on the top of my screen. Okay. So stealing 2,3-BPG from PGM would kill the enzyme. The active site is not back to the start. The solution is we siphon it off from 1,3-BPG and then use an alternate path or enzyme to do the chemistry. So, and I think these are the same exact words that you guys have for your homework. So the purpose of the enzyme hexokinase is it's step 1 in glycolysis that converts glucose to G6P. So for a cell that is hexokinase deficient, okay, when you are deficient, and we talked about this when we did these slides, anything that's deficient means you are going to build up metabolites prior to it, and you're going to have a deficit of any enzyme or any metabolites after it. So if we are talking about somebody who's hexokinase deficient, we're going to have low levels of 1,3-BPG, which means low levels of 2,3-BPG. Thumbs up if that makes sense. Okay, so thinking about then pyruvate kinase. Pyruvate kinase is the 10th step of glycolysis. It interconverts phosphenolpyruvate and pyruvate. So again, per above, since we are pinching after the pathway, or our pinching happens after 1,3-BPG synthesis, that means 1,3-BPG is going to accumulate, which also means 2,3-BPG can accumulate. Okay, so part of putting these pieces together with this plot is saying, well, first of all, I need to know what these enzymes do. Okay, I need to know what hexokinase does. That's the first enzyme in the pathway. Walking through the logic, that means I'm going to be having a pinch in my pathway right at the beginning. So I'm going to have low levels of 1,3-BPG, low levels of 2,3-BPG, which means I'm going to favor hemoglobin in the R state, which means I'm going to have tighter binding of hemoglobin, which is what we see here. That curve shifts to the left. Remember, this is a binding curve. Okay, this is a binding curve showing the saturation level of oxygen to hemoglobin. Everybody good with that? flip side of
Can
that
you say
is
that again?
Yep. So when we have an enzyme that we're pinching off at the beginning of the pathway, we went through the logic. Hexokinase deficient cells are going to have decreased levels of 1,3-BPG because we're pinching before we're making 1,3-BPG. Okay. We know that we make 2,3-BPG from 1,3-BPG. So that means we're going to have low levels of 2,3-BPG. Now we need to map on what we know 2,3-BPG does with hemoglobin. Okay. Hemoglobin with low levels of 2,3-BPG is going to be more in the R state. R state hemoglobin binds oxygen more tightly. When we bind more tightly, our curve shifts to the left. Good now, Brenna? All right. Now it's really just the flip side. All of those up arrows become down arrows and vice versa when we talk about cells that are pyruvate kinase deficient. Now we're pinching all the way at the bottom of the glycolytic pathway. So everything accumulates prior to it, which means we have higher levels of 1,3-BPG, higher levels of 2,3-BPG. That means high levels of 2,3-BPG means that we are going to be favoring the T state of hemoglobin. T state of hemoglobin means I have a higher P50, I bind oxygen less tightly, however you want to word it. My binding curve shifts to the right. And that's exactly what we see here. Okay, so does that make sense? So I want you to be able to think about two kinds of curves that we have here. This was a binding curve that we saw in this later question. Okay, let me just make sure. I think that was the last sort of question here. So your last question says, you know, based on that data above, explain what we see in this binding curve. Okay, so binding curves that we see here, but we see similar kinds of curves with an earlier question. Let me find it here, where we talked about this. So this is still sort of pseudo a binding curve, but the binding curve really relates to doing chemistry on the substrate. We make the assumption that if we're binding our substrate, we're doing chemistry on it. So we're relating that substrate binding to doing chemistry with that substrate. So the same kind of thing, but now our y-axis is enzyme activity rather than just binding. Okay. All right, so that got us through our homework. Let me come over here just for a second, and I want to make sure, because I'm pretty sure I included these slides here. Okay, I would make sure, we didn't really cover this in class, but because there are mechanism questions, okay, this will be a good opportunity to kind of go through, and I'm happy to take a little bit of time right here. We still have some time left. Oh, my watch died. What time are we at? Let's see. Oh, my phone died too. I think
It's 9:53.
9:53. Okay, so we have a little bit of time here. So let me just kind of recommend what I would do, show you how these slides are set up, and maybe just go through and review them for yourself. I'll have to go and look and see if I have a video of me walking through them. I can't remember if I do. So how these are set up is they start out by just looking at the mechanisms that you guys had to do. Like if you just had a BRM that you did last semester, you know, a reaction mechanism, one of those 22 that you turned in. So this was hemiacetal or hemiketal. In this case, it's a hemiacetal going to an aldehyde or ketone. So to do that, that means we're actually going to be breaking this bond here. So how we do that is we're going to protonate this oxygen. We always protonate things because it makes them better leaving groups. So this internal nucleophile here is going to push that good leaving group out. I create a very electrophilic intermediate here. And then, well, actually, we're not doing additional chemistry here, but we've got then this proton that's going to be removed by a base, and we get back to our carbonyl. Okay. So that's actually how we do ring openings. So if you want, if I flip to applications to glycolysis, and whether you do these one at a time or you do look at all of the review ones and then you come here. So when we talked about, let me come back here just for a second. This was related to how we do ring opening. So we're going to protonate an oxygen here. that's going to make a carbon-oxygen bond more likely to break. So if you come over here and we see applications to glycolysis, oh, hey, look at this here. I protonate an oxygen that makes a carbon-oxygen bond more likely to break. And that's how I do ring opening. And then I've got this carbonyl here that's protonated. I use my base to remove that proton. That's the chemistry for doing ring opening. Okay. When we had hemiacetal or hemiketal going to aldehyde or ketone, okay, that was how we do ring opening. So this just kind of shows you that same chemistry, but now mapped on a structure where that happens in glycolysis. Okay. So if you want to go through and memorize all of these, I will tell you one of these is on there. Okay. Which means you need to know the structures of things. So it's your call if you want to just memorize all of these, or if you're like, I see the patterns, I see nucleophiles and electrophiles. Okay. You are going to see one of these that you have to do. Okay. And it will be not necessarily the way that you saw it last semester. It's going to be the application. Whoa. Don't, I'm in the middle of a class.
Dummy, I didn't do anything.
But you're scaring her. You're doing hide and seek or something with her.
Just said hi.
No, you're doing your little like,
I didn't do a damn thing.
she thinks you're playing hide and seek with
What's the
her.
matter? She's a dumb.
Don't call her dumb. Don't call her dumb. All right. So you're going to see one of these applications to glycolysis. I'm going to ask you one of these couple of mechanisms. So I guess there's 1, 2, 3, 4, 5 possible mechanisms. I will tell you that it's one of those. Okay. And then there's another one that you sort of haven't seen, but I want to see, can you like put stuff together? Okay. So we got a couple more minutes. Any other questions? Anything else that I can go over for you? I'm going to stop sharing my screen for a second. I'm going to go back to my exam. And I'm just going to flip through it and make sure that I've touched on everything. So yeah, there's that like laundry list of, yeah, I do get into the weeds with glycolysis. Okay. Then there's this section on beyond glycolysis. Okay. Which is some of the bigger picture questions. And then here's this fictitious metabolic pathway question. There's only 4 pages. Okay, so it's like exams like you've seen before, pretty much those first 2 parts where it's like just single letter question or single letter answers is about 1.5 pages. And then I have a couple of short answer ones that basically are homework questions. They're exact homework questions. And then the second 2 pages are stuff that you haven't seen. There's answering questions about a fictitious metabolic pathway. And then draw out a mechanism that shows me. And then this is one of those 5 that I told you it's going to be. And then a mechanism that you sort of haven't seen, but can you extrapolate what you know onto a new system? So that is the exam. So Brenna, I still have to think about it. I will get you an answer. I will think about it over the next little bit here. I will post an answer in Teams. But let me ask, since I guess everybody except for Megan is here, who has a class at 8 a.m.? Okay. So because people have classes at 8 a.m., that's just the only concern, is it's a little bit of an unfair situation if I sort of let somebody come in a little bit early. So I think for that reason, Brenna, I think I'm going to have to say, we're going to give it the, and you guys always know I'll let you stay as close to 10 o'clock as you want. Who has a class? I think some people have class right after, right? Okay. So do your best. Because I don't want to be unfair and have some people have more time than others. But we'll start right at 9. And then if Brenna, the first thing that you want to do is I'll even let you have a blank piece of paper if you want to just kind of like, as soon as time starts, you can write down whatever you want to write down to kind of help with that pathway. Other questions?
I just have, I missed it. So for the pentose phosphate pathway,
Mhm.
the first step is good for glutathione regeneration and lipid what?
Biosynthesis. Yep. We could also just say, and we talk specifically about lipids, but anytime that we need, you think about electrons as glue. So anytime you need to have glue to make bigger molecules, those are anabolic processes. We often see NADPH for that. But the classic example that we're going to spend some time talking about is lipid synthesis. Anything else? Elizabeth, you look like you are about to fall into your computer.
It's been a rough day.
Yeah. Yeah. And I know I don't make things easier. This is rough stuff. Biochemistry is not an easy course. I know that. But keep in mind, the hope is that there's plenty of stuff, whether it's homework or case studies, like you guys get full credit for just participating in doing those. There's no like assessment of did you do it right or wrong. So there's lots of opportunities for getting stuff right so that my exams are a little bit on the harder side. I know that. Oh my gosh, look at this girl. Yeah, I'm talking about you right there, Charlotte. She's really bad right now, though. She's not eating. I don't know if I told you guys this. I think it's because she had rice and chicken. Like right after she had her surgery, she had some like diarrhea right afterwards. And the vet said, you know what? Put her on a bland diet. Just give her rice and chicken. She loved that. She wolfed it down. And then we transitioned her back to kibble. And it's been like 3 days. She really hasn't eaten. Like she'll eat treats. She'll eat like macaroni, a bowl of macaroni and cheese that fell on the floor. So like she's hungry. She just does not want to eat her kibble. And look at her. She's like, you're killing me, Mom. I'm so pathetic right now. I'm like, no, I'm going to call your bluff. It's like the kid who refuses to eat their vegetables. It's like, no, you can have that for breakfast the next day. You don't want, if you're not hungry for breakfast, fine, we'll have it for lunch. You're going to eat your vegetables before you eat something else.
Dr. Prenny,
Yeah.
you have to, what my parents do is they throw tuna in, like the cans of tuna, they'll split it for both of our bulldogs. And so they get like the oil from the tuna and they kind of like smells it a little bit and they just woof it down. But sometimes my dog Daisy, she eats it too fast and then she just throws it all up.
What we did when we transitioned her back to kibble, I still put like some of the, and it was canned chicken that I was using because I'm like, I don't have the time to cook you a chicken breast. I'm just going to use canned chicken.
It's like putting broccoli in Jello.
So yeah, no, she would. And then we tried to like hide the chicken like underneath her kibble and she would literally like nose all of the kibble out and then eat. And she's bad. So
yeah, she's a smart dog.
She'll get hungry enough. She'll eat. So. All right. Well, if you think of anything else throughout the evening, if you want, you can send a message via Teams, but I'll just plan on seeing you guys tomorrow morning and we'll have it ready right for you to start at 9. Okay. And then as soon as this is done, it takes a few minutes for the recording to get posted to my channel. So as soon as I have that link, I will post it in Teams if there's anything you wanted to go back and see again. Oh, and I will also post, I'll probably just do my copy. You may have to search around a little bit for where those answers are, but I'll post that for you guys. The only thing that I would respectfully ask is that it's not something that sort of floats out there. I will make that available for you guys, but I think I just don't want it out there for students for next year. That's my only request. All right. See you guys tomorrow. Good night.
Hey, cheers, Dr. Prenny.
All right, we'll see ya.